Hi, everybody. This is the second video for the third week. I'm going to move us over to my PowerPoint and also spend some time on the whiteboard. Today, we're going to talk about viral genomes. We talked a little bit about them briefly in the first lecture for this particular week. And I also talked about genomes and DNA replication and protein synthesis in week 2. In this particular one, we're going to focus just on the viral genomes. We'll start with listing the different types of genomes that can be found in viruses. I will also explain how the genomes which genomes have the same number of protein synthesis steps as we would see in cellular DNA. I'll briefly review cellular DNA protein synthesis so that you can refresh your memory of how that works. Then I will expect you to know which genomes have more steps for protein synthesis than we would see in cellular DNA, and which genomes have fewer steps. Viral genomes are smaller than what we would typically see in a cell. Most viruses have less than 300 genes. The goal, remember, of viruses is to get in quickly into a host cell, make viral proteins, and copy their genomes to make new virions to then send out and infect other cells. The viral genes will encode for several different products, the capsomere proteins, and often structural factors like spike proteins. They will also encode for enzymes that are needed for viral replication in those viruses that need them. The viral genomes can be either RNA or DNA. This is in contrast to cellular or cell genomes that are only DNA. The viral genomes can be either single-stranded or double-stranded. They can be single or segmented sections, and they can be either circular or linear. Whichever type of genome a particular strain of virus has, all of those virions will have it. But we know that, for example, influenza versus measles may have a different type of genome. The genomes of viruses can be classified by the Baltimore classification, and this breaks viruses into 7 different classes. At the top, we have double-stranded, single-stranded DNA viruses. Then we have double-stranded RNA viruses, single-stranded RNA viruses that are either what we call a positive sense or a negative sense strand, and I'll explain that in just a minute. And then we can have 2 different types of reverse transcribing viruses or retroviruses. We can have either RNA or DNA reverse transcribing viruses. With that, what I'd like to do is move us over to the whiteboard and go through kind of the steps of protein synthesis and how each of these genomes will dictate how many steps are required. So on my whiteboard, what I'd like to start with is start talking first and kind of review what we did in week 2 of protein synthesis. And remember, I also said this could be called gene expression. They're interchangeable. In a cell, the way that this would work, I showed you that there would be the DNA, and I drew it kind of as a ladder, right? That double helix that we've unwound and kind of straightened out. We have the base pairings that act as the rungs of the ladder. And if you remember, I said that you could imagine this going through the top of your computer screen and down through your floor. Within that, what we would expect to see then is that along the length of that, there would be areas that would be codes that code for a particular product, and we call these genes. So there would be genes all the way up to your ceiling and all the way down to your floor. And a gene is just a specific code that when it is transcribed in the cell, so we would go through transcription, what ends up being made, or the product, would be mRNA that has that code from the gene. That mRNA then would go to the ribosome, and it would go through the step of translation, which reads the mRNA and ultimately produces a protein, which is our ultimate goal. So hopefully you can see here that this takes two steps in a cell, right? We have transcription and we have translation. Oops, sorry, put a 1 there. It's meant to be a 2. What I'd like you to do is think about that as kind of the basis for protein synthesis. And then we'll talk about these different types of viral genomes and what steps they would have to go through in the host cell in order to make the proteins they need for their capsomere or their capsid. Keep in mind also, though, that their genome will also be replicated in order to make virions that have all the proteins and the genomes to be released. So if we think about the viral genomes, I'm going to shorten that list that we had on the slide into a little bit different categories, but you can think of them as the similar, but I'll put some of them together. So the first kind that we can have is a double-stranded DNA. The second is we can have single-stranded DNA. Then we can have our double-stranded RNA. I'm going to say our positive sense single-stranded RNA, our negative sense single-stranded RNA, and then I'm going to bunch together our DNA and RNA retroviruses and just put that as retroviruses. So the first ones that I want to think about is the ones in blue. So double-stranded DNA, single-stranded negative sense RNA, and our double-stranded RNA. The ones in blue all will go through the same steps. They will look just like the cellular. They'll do transcription and they'll do translation. So these ones have the same amount of steps as we saw with cellular protein synthesis. So write those out, and this one is the same as well. So we do the same steps. They have to transcribe it into a mRNA, and then that needs to be translated at the ribosome. With the other ones, I have a couple of other ones that are of interest to me. With this positive strand, single-stranded RNA, the really cool thing about this one is the positive sense RNA actually looks just like mRNA to the host cell. And so this one gets to skip transcription and it goes right to translation at the ribosome and makes that protein. So this one's a special one because it actually skips a step or takes a shortcut. And then in red, I want to go over the ones that would take an extra step. So in both of these that are in red, they actually have to do something called reverse transcription. And then they can go on to transcription and translation. Sorry, I'm running out of room. Try to make it a little bit better down here. So we have reverse transcription, which means we actually make another strand or 2 strands of DNA. Then we do transcription when we need to make the protein. And lastly, translation. So the ones in blue have the same number of steps as cellular protein synthesis. The ones in green skip a step or take a shortcut. And then the ones in red have to add a step. And this is kind of thinking about the time that it would take a virion when it gets into the host cell to make the proteins it needs to make the capsid and the spike proteins. I'll go back and review this on our PowerPoint, and hopefully that will be clear. If you have any questions, I know this is a little bit complicated, please don't hesitate to reach out to me. So the viral genomes, depending on what they start with, that determines the steps that have to occur not only to replicate the viral genome, but in the case of what I was just showing you, of how to synthesize the necessary proteins. So here's the list of what I had. The same steps or same step number as cellular DNA would be our double-stranded DNA, our negative sense single-stranded RNA, and our double-stranded DNA. The genome that would take less steps in order to make proteins would be our positive sense single-stranded RNA. And then lastly, those retroviruses and single-stranded DNA actually take on an extra step. They have to go through reverse transcription before transcription and translation. This is the checking in. I will pause here. You can stop the video, write down your answers or say them out loud, and then turn back on the video for the answers. The first question is, explain why the genome dictates how many steps are necessary for replication. The genome, depending on whether it's single-stranded or double-stranded, or it's a retrovirus, actually has different starting material, and so it dictates how many steps we'll need to do replication and to do protein synthesis. The second question is, which type of virus reverse transcribes its RNA genome into DNA? The answer to that would be retroviruses. And in this case, because it's RNA, it would be RNA retroviruses. The third question is, which type of viral genome can immediately go to translation of proteins? The answer for this would be the positive sense single-stranded RNA. Thanks, and let me know if you have any questions.